Good morning to all our viewers in Europe and a good day to all our viewers in other parts of the world. Thank you very much for taking the time today to join our webinar on abbreviated new drug applications, in short ANDAs, for certain highly purified synthetic peptide drug products that refer to listed drugs of rDNA origin. I'm very much looking forward to the presentation of my colleague here about Bachem's capabilities to address the new FDA guideline. Before we begin, I would like to give you a brief introduction. Bachem is a global CDMO and innovation-driven industry leader in the development and manufacture of peptides and oligonucleotides. We celebrated our 50th birthday just a few weeks ago. We are well known for providing high-quality products for various applications, including research, clinical development, and commercial pharmaceuticals, as well as diagnostics and cosmetics. We operate internationally. Our corporate headquarters are located in Bubendorf, Switzerland. And we have locations in Europe, the US, and Asia. Bachem is listed on the Swiss Stock Exchange, and for those who are interested, more details about the company can be found on our webpage. I would like to take the opportunity to introduce our speaker for today, Dr. Sergei Malashikin, who is a group leader, generics and business development at Bachem, Switzerland. In this position, together with his team, he is responsible for customer projects with complex generic peptide APIs manufactured at Bachem, as well as for early and development stage generics projects. Prior to joining Bachem, he worked in a commercial role in a Swiss life science company and in R&D of a Japanese technology corporation. He earned his PhD in organic chemistry at the University of Zurich, Switzerland. I would also like to take the opportunity to introduce myself. My name is Martin Ostermeier, and I'm product manager for research chemicals and specialties at Bachem since September 2019. After my PhD at ETH Zurich, I was scientific officer at the Paul Scherrer Institute, where I was author of 10 scientific papers and inventor of 3 patents. At the same time, I was CEO, co-founder and chairman of the board of directors of Interx Biotech, where I transformed a business idea in the field of artificial intelligence for drug discovery into a commercially successful company with international customers. Now, let me guide you through our first poll. The question I would like to ask our viewers is, do you work on generic drug development using synthetic peptide APIs? So most of you said they have no experience so far. That's interesting. So I'm glad to have you here, and hopefully you find this webinar very useful. We will continue with our introduction of Bachem in general and as your partner for generic APIs. Okay, great. As I mentioned earlier, Bachem just completed 50 years of business. In this pie chart, you see our 3 business sections, which are research, chemicals and specialties, commercial API, including lifecycle management, and chemistry, manufacturing and control development. With that business model, we serve customers from various markets globally, including pharmaceutical and biotechnology companies at all development stages, as well as research groups, both in industry and academia, and diagnostic and cosmetic companies and others. We offer products and services in all stages, from research over development to commercial application. In today's webinar, we will focus on generic APIs. We are located at various parts of the world globally. As mentioned earlier, our corporate headquarters are in Bubendorf, Switzerland, close to Basel. The second site in Switzerland, in Vionnaz, specializes in large-scale manufacture of small molecule APIs, as well as FMOC amino acid building blocks. We also have a site in St. Helens, UK, which specializes in the development and manufacture of research-grade peptides and products for use in vitro diagnostics. The UK site has been certified for the manufacture of peptides used as raw materials in medical devices. In the US, we operate under the Bachem Americas umbrella. The Bachem Americas operations are based out of Torrance, California, which is just south of Los Angeles. We have a secondary site in Vista, California, which is near San Diego. In addition, we have a sales office in Tokyo, Japan. Since we recently celebrated Bachem's 50th birthday, let's go back briefly in history so that I can tell you how innovation has been and still is the driver of our success. In 1971, Bachem was founded 7 years later. Bachem was the first CMO to produce a peptide API, which was thymopentine. 20 years later, the company has produced the APIs goserelin, calcitonin, and aprotinin and went public in 1998. In 2013, Bachem produced interferon beta-1a. With this example, I hope I have demonstrated you how Bachem continuously increased manufacturing capabilities from small peptides to such with more than 150 amino acids. Today, we have implemented molecular hiving used for peptide synthesis and continuous chromatography used for product purification. For those who are interested to learn more about these techniques, please visit our webpage. Beyond that, we do have an innovation management internally to drive innovative ideas at Bachem in all areas. Regulatory requirements are increasing in complexity from year to year. Today, we are showing you what Bachem brings to our customers in terms of dealing with the new ENDA guideline so that our generic drug products with synthetic origin are compliant. With our long history, we have become a leading large-scale manufacturer with 24 generic peptide APIs in our portfolio currently. With this webinar, we are going to provide you more information on how to comply with the new FDA guideline for synthetic peptide drugs. Just some housekeeping before Sergey begins. We do have a Q&A function. You should be able to see the Q&A button at the bottom of your screen. Please use that to input any question that you may have at any time. And at the end of the webinar, we'll have time for questions and we'll do our best to address as many questions as possible. And with that, I would like to turn it over to Sergey.
Good morning, everyone. Good morning, Martin. Thank you for your kind introduction. And let me begin with a presentation. So we called our presentation, our webinar today, Bachem capabilities to address the new FDA guideline. So called it new, but the final version of the guideline was published a month and a half ago, and the very first draft has been made available to public in October 2017. This guideline is part of the US FDA initiative to simplify the approval pathway for generic drug products and improve consumer health access to affordable drugs. And although the guideline is explicitly listing 5 peptides, it can be applicable to a large number of peptides now and also in the future. So peptides, as you probably know, lie on the interface of small molecules and biologics, and FDA defines a peptide as an amino acid polymer with 40 or fewer amino acids. And certain peptide can be produced by rDNA technology or synthetically, and Bachem produces its peptides synthetically. And the regulatory pathway for peptides can be challenging. So FDA recently has placed a special focus on complex generic products, which have been now defined. And this is a long list of drug products that fall under this definition of complex generics. For Bachem, the most interesting part is peptides, because it's the company is a peptide manufacturer. Now, if we look a little bit into the situation in the past with generic products that refer to the original drug that contains an API of recombinant origin. The fact is that FDA, up until recent, did not approve a proposed ANDA for synthetic peptide that is a duplicate of a previously approved peptide of rDNA origin. So the agency was concerned about difference in the purity profile and hence safety of the drug with generic drug to the patients. And this led the agency to a dilemma. On one hand, they wanted to ensure that affordable drugs are approved on the market, but on the other hand, the patient safety cannot be compromised. So what has changed in the last decade? FDA believes now that the current state of technology for peptide synthesis and characterization allows ANDA applicants to demonstrate that the active ingredient is the same as in the previously approved drug, and in addition to the same dosage, form, mode of administration, conditions of use, etc. So progress in technology is allowing now to demonstrate the sameness and prove that the drug is safe to the patients. Now, sameness is a multifunctional term, and this sameness is defined in the guideline, and the number one comes sameness of active ingredient, and this is physical, chemical, and biological characteristics. It also involves sameness of impurities, impurity profiles of peptide-related impurities, oligomers, aggregates, and together with the impurity, it all becomes immunogenicity of this potential new impurities or impurities that are at higher level. So this risk of immunogenicity does not differ from that of the RLD. And finally, biological activities, they should be also the same as the RLD. Now, a crucial factor in determining whether this peptide API is appropriate for approval as an ANDA, it's the sameness of peptide ingredient, of the active ingredient. And this is determined by applying orthogonal methods to conduct physical chemical characterization. If we go through the list that the guideline outlines, we see that the primary sequence should be defined. We do it routinely, and this comes from evidence from the synthesis. We confirm it by amino acid analysis, by GCMS and chiral GCMS, by mass spectrometry, and finally by correlation with the reference standard. Then the secondary structure, this is a bit more difficult to confirm, but still we use instrumental techniques such as CT and NMR spectroscopy. And then oligomers and aggregation states, there is a number of methods that can be implemented. In most cases, this is a combination of methods: filtration test, thioplasmin test, CD spectroscopy, and seismic fusion chromatography. And finally, biological activity or bioassay is performed. Now, whether peptide is appropriate for submission as an ANDA is determined by its impurities profile, and certainly all peptide APIs, they contain certain impurities irrespective of route of synthesis, and these impurities result from manufacturing process or from degradation. They are decisive for determination if this peptide is suitable for submission as an ANDA. So in order to make this impurities profile comparable with the original drug, peptide API, synthetic peptide API should meet the following criteria. So the applicant or the peptide API manufacturer should confirm or verify that the level of each impurity related peptide is the same or lower than the RLD. They should reduce or mitigate the risk of related impurities, and this can be achieved by comparing the synthetic peptide with that of the RLD, with the original peptide drug. No new impurities should be at a level above 0.5%. This is the acceptable batch variation that FDA believes to be safe. any new impurity is characterized and specified if this impurity is above 0.10%. The applicant or the API manufacturer should justify why such new impurity does not affect the safety of the drug, and any unspecified, if it's an unknown impurity, it should be controlled at the level below 0.10, and impurities shouldn't change aggregation behavior of the synthetic peptide. So also immunogenicity, in addition to this immunogenicity and chemical properties, physical chemical properties of the API, biological properties should not alter. And this is defined by bioactivity of the peptide API and the final drug, the immunogenicity and the toxicity, possible toxicity of impurities. Now let's have a closer look at take a glance at the history of approval of original drugs listed in the guideline. And if you look at this approval dates of peptide drugs on the US market by the FDA, you clearly see that the development of generics started well before the first drug guideline was issued in October 2017. So now if we have a closer look at what influences the impurities profile of the final peptide API and hence the impurities profile of the drug product, generic drug product, we see that the highly pure material, protected amino acids and building blocks, they are extremely important to achieve excellent purity of the final peptide API and ensure lower content of de-isomer in the final peptide. We also, is important, the optimized synthesis and in-process control with type criteria, the purification of the final drug of the crude material, and the QC release and control with type limits. So now, how to control the purity of the starting material, which is, as we saw, crucial for the purity of the final material. We use validated QC method to control. We set type limits for related impurities for all starting material, including stereoisomers. We have specified individual impurities and set limits for unspecified impurities also for starting material. And some of the starting material, the key starting material, is manufactured in-house at the company. If we now talk about synthesis and optimization of the synthesis, I have here a table that shows, so the peptides that are listed in the guideline, they are on average 30 amino acids long. And in order to get the usable purity of the crude material before it goes on the HPLC column, in order to achieve even 75% purity of the crude material, we have to reach the 99% yield on every coupling step. And this is not a trivial task. This is an example of an API that Bachem produces and that is listed in this guideline. And you can see that after the optimization of the synthesis, we increase the purity of crude material from 27% to 76%. And this is important both for economy of process and for the purity of the final API. And this means that indeed we have achieved 99% average yield on every coupling step to arrive at this purity of the crude material. I'm going to guide you through two practical examples. One of them is a glucagon. It's a synthetic peptide API that Bachem is manufacturing for quite a couple of years. And its manufacturing process was developed well before the guideline was issued, with some later optimization and a lot of analytical work to allow this NDA submission. So if we again go through the guideline says that one of the key and difficult criteria to meet is any new impurity should be characterized and specified. In order to know which impurity is new, we have to compare our glucagon API with the original drug, or let's say drugs, and see which impurities are new, not present, or present at a higher level than in the RLD. And these impurities have to be specified. Now, if you look at this picture, you will see that how the peptide API, the glucagon API looks like, and all the impurities that have been unambiguously specified first by MS experiments, then by sequencing, and finally confirmed by spike testing. So we synthesized all these impurities, including 28-point isomers, and confirmed their presence and their concentration in the API that we offer to our customers. So once we've done this tremendous analytical work, we have to specify all impurities, set limits, and we set these limits based on three factors: comparison with RLD, so we know which one impurity at which level is present or absent in the RLD, identification of these impurities, and then batch record. So when we set the specifications, we have to be sure that we can control these impurities at this level and supply consistently high quality API, and this will not raise question during approval and commercial phase afterwards. Sorry, I have to go back for a number of impurities, really few of them. We had to conduct immunogenicity studies because the guideline says if this impurity is new and the level of this impurity, the concentration of this impurity varies between 0.10 and 0.5, we have to justify its limits, so prove that this is safe. And then for glucagon, we have conducted immunogenicity studies for these few impurities in collaboration with an external service provider, and then prove that these impurities have negligible immunogenicity risk. Now, if we have a look at the second case, and this is a peptide API that is now being developed at Bachem, and the development started when the guideline, the first draft guideline was published, so we knew what the requirements are. I can't show you a lot, but what I can show is the comparison of this newly developed peptide with the original material with RLD, and then it's evident that the synthetic material is much purer than the recombinant material that is present in the RLD. And if I show you the analytical certificate, you will clearly see that only a few impurities are at a level that requires attention, so above 0.10. And also, there is one or another de-isomer that is of concern to us, but we are currently addressing this problem. And then once the process is fully validated, we'll be in a production to support the NDA filing with this peptide. FDA also pays attention to aggregation behavior of peptides, which is a common problem when making peptides, and there are certain ways how to overcome this aggregation or control it during manufacturing and purification. And the guideline says that the aggregation of the synthetic peptide should be pretty much the same or better aggregation behavior than that of the RLD. So it is a common problem, it is a known problem, and it would, covering this topic would require an additional webinar or a presentation. But the methods to measure quantitatively and qualitatively control this aggregation are known. We have experience with all of this method and are perfectly good to do so. And we do control aggregation of synthetic peptide APIs that we manufacture either during manufacturing process and purification or after that, or both. Meeting this NDA guideline requirements is indeed challenging. They are clearly defined, but it requires or implies, supposes a close collaboration between Bachem and our customers, NDA applicants. And in order to provide the support and encourage this close collaboration, we have a dedicated RA team that is experienced with such type of submissions, both on the substance side and on the support of our customers with this submission. Bachem keeps investing in the existing products and in the development of new products. So we pay a lot of attention to the lifecycle management, to maintenance of the products, and we have teams, dedicated teams, led by a professional project manager, and every team is composed of subject matter expert and is dealing with that individual API, peptide API that we offer to our customers. And this allows us to always be at the forefront and products to remain and be compliant in the future. So we can conclude now from the multiple years of experience with this guideline and even before this guideline that it is indeed applicable to the peptides that Bachem manufactures. We have collected a broad experience with this guideline and in supporting customers in communicating and convincing FDA. Indeed, synthetic peptides can be manufactured in consistently high quality to meet the guideline requirements. It is well possible to identify, specify, and control impurities with tight limits to meet again the requirements that listed in the guideline and show the sameness and control strategy for sterile isomers is developed, implemented for certain APIs where this topic is of concern. And also know how to justify impurities limit by conducting immunogenicity or toxicology studies, and we have done this for certain peptides that we offer to customers. Based on this knowledge and analytical work and great batch record, we can compile a comprehensive dossier. These dossiers have been filed with FDA under super requirements, and these dossiers are available to our NDA customers willing to file their drug products on the US market. So I'm approaching the end of the presentation. I would encourage you to have a look at our homepage or get directly in contact with us. Hopefully we will soon be able to meet on the CPHI or any other event in person, but for now, please follow us on the LinkedIn or drop an email if you have any questions. I would like to conclude by saying a big thank you to my colleagues from different departments, QC, QA, R&D, production, sales, of course, for excellent work, their support, for sharing their knowledge, and for just being a great team. And I would like to especially thank my colleagues Gerhard Haas and Thomas Ziegler, who gave a really valuable feedback when preparing this webinar. Thank you very much.
Thank you, Sergey, for the presentation. I see we have a couple of questions, but there is another poll. And the question I would like to ask you is, did you come across the new ENDA guideline in your projects already? So again, you should see the options pop up on your screen. And these options are, yes, I deal with it, no, but I heard about it, and no, not yet. So please choose the one which is applicable for you. and I will give you real-time results after 10 or 20 seconds. In the meantime, I would like to thank you for your time, to everybody who contributed to the polls, and I'm looking forward to the result. Okay, so we have yes, I deal with it, and no, not yet, equally contributing in the results. That's interesting. Now let's go on to the Q&A. So we have a couple of questions. If your particular one didn't get answered, always feel free to email at webinar@bachem.com. And now let's start with the first question. In your understanding, are the concepts expressed in ENDAs for certain highly purified synthetic peptide drug products that refer to listed drugs of rDNA origin guideline applicable to any other peptide active ingredient than liraglutide, glucagon, nesiritide, teriparatide, and teduglutide?
Yeah, thank you for this question. I've asked it. It's indeed a good question. When the guideline appeared, there was at least one candidate that was expected to be included in the guideline. It's the next generation of GLP-1 analog semaglutide. So it should definitely be applicable to this peptide. But we also see from feedback that we get from the agency for other peptides, for really synthetic peptide that refer to the original synthetic peptide as well, that similar questions are asked without mentioning this guideline, questions about transferring of impurities, conducting immunogenicity studies to justify these limits. So it seems to be a trend, but let's wait a little bit until it materializes in a documented guideline.
Thank you, Sergey. So here is another question. The guideline mentions evaluating the peptide-related impurity profile in the proposed generic synthetic peptide in comparison to the RLD. This would imply comparing a drug substance, it is proposed generic synthetic peptide, to a drug product, it is RLD, thus excluding the drug product process or excipient-related impurities. Is that correct? Please, could you comment?
This is also a reasonable question. Indeed, the best way would be to compare substance with substance, formulated drug with formulated drug. So FDA listed all these requirements nicely and in an understandable manner, but I forgot to mention how to get access to the recombinant API that is used in the original drug. This is much impossible. So we have to go around this, and in most cases, it is well possible to rely on just the drug product that is available on the market and either directly identify impurities in it. Sometimes some is needed to get rid of excipients and then that interfere or might interfere with the results of the studies. So we did this for a number of APIs, and this worked well. It's a little bit more complicated when you try to compare study aggregation, but even it's possible to overcome this issue.
Thanks, Sergey. Here's the next question. What is the filtration test for characterization of oligomerization/aggregation?
And this is a quantitative test that proved to be working well, and it shows the velocity of filtration through a filter. And then if the peptide API, synthetic peptide in solution forms aggregates, then or jellifies, then you see it immediately in the filtration test, and this is a red flag, meaning that attention is needed. And then it's decided on an individual basis what to do, whether further measurements are needed or not. So it's a quantitative test, but it's a quite reliable test for certain peptides that we could use.
Here is another one. For toxicology studies, which animal model study generally will be used?
Well, this is defined individually for peptide depending on its level. But what we've done, we also did it in collaboration with a third-party service provider because we don't have facilities to do this. This was animal rats, unfortunately. Wherever possible, we try to put such experiments, but toxins, yes.
Here, another one. What do you do if one or another impurity cannot be separated with the release method? Do you use multiple release methods?
This happens. This happens also with stereoisomers. We try to avoid using multiple methods wherever possible because it makes the release more complicated both for us and for our customers. But sometimes it is needed, or we can control certain impurities at the in-process control stage, and this is sufficient to control this level and justify that this impurity is not present at a level that is not permitted.
Okay, thank you. Do you routinely control high molecular weight impurities in peptide APIs and specify them?
We routinely control high molecular weight impurities if it is relevant for a certain peptide manufactured using a process that was developed here. We control them using various methods, either such quantitative methods, for example, this filtration test, or we have also more quantitative methods like size exclusion chromatography, both for covalent and non-covalent activities. So we do control this activity, yes.
Okay, and let's finish now with the last question. How do you verify the level of stereoisomers in a final API?
Yeah, this is not always trivial task, so we use chiral amino acid analysis. This, however, might lead to some overestimation of isomers present in the final peptide. So if this doesn't work reliably, or we can't get reliable results, we, in this case, also implement a more comprehensive technique, such as photolytic technique, where we treat the peptide with a certain enzyme, this impurity, and then establish the concentration of fragments, and then derive back the concentration of the isomers.
Thanks a lot, Sergey. So I think that's it. We answered a couple of questions. If your question wasn't answered, then please feel free to email us at webinar@bachem.com. For more information, please check out our webpage and our social media channels. We thank you for your time. I hope you have enjoyed the webinar, and we say goodbye. Have a great day.